I think we'll go on for the second talk, and shifting gears, we'll go to the back side of the cornea, and we invite Dr. Olga Antonova to deliver her talk titled Transplantation of Native Corneal Endothelial Cells in an In Vitro Experiment.
Good afternoon, everyone. And first of all, I would like to thank Indian colleagues for the invitation, and it's a pleasure to speak in your professional society. And I will start my report. Today, there is only a surgical option for the treatment of endothelial corneal dystrophy, which include Fuchs corneal dystrophy and pseudophakic keratopathy. Among keratoplasty techniques, in addition to classic penetrating keratoplasty, Distemate stripping endothelial keratoplasty using a microkeratome or femtolaser and transplantation of Distemate's membrane with endothelium are most often performed, and less commonly transplantation of a fragment of Distemate's membrane, for example, hemidemic or quarter-demic, is performed, or the donor endothelium is not transplanted at all, and only Distematorexis is performed. But despite the high results of these surgical techniques of keratoplasty, they have some disadvantages. For example, damage to the donor graft during preparation or during its transplantation, and also detachment of the endothelial graft in the early postoperative period. These problems initiated the search for ways to isolate and cultivate corneal endothelial cells, which is still relevant today. In 2014, Japanese scientists transplanted cultured corneal endothelial cells using a ROCK inhibitor and patients with pseudophakic dystrophy. A required condition was a forced hand-down position for 3 hours, and they showed results in follow-up periods of 2 and 5 years postoperative period. And this slide shows photos of all 11 eyes before cells injection and 2 years after. 5 years after surgery, normal corneal endothelial function was restored in 10 of 11 eyes. It should be noted that in Russia we have a law that restricts the use of cultured cells products. And the restrictions are very strict, and we can say that this law practically prohibits the use of cultured cells. That's why we have developed a method for getting a suspension of native endothelial cells. These techniques involve the primary separation of a roll of Distemate's membrane with endothelium. Then the Distemate's membrane is placed in a solution of chemically stable trypsin, and after that, using the thermoshaker device, endothelial cells are knocked off the membrane. The cells settle at the bottom of the tube, and then we washed the cells from the enzyme solution, and in this form they can be transplanted. On this slide you can see the result of confocal microscopy with staining of living and dead cells, and there are obviously more green living cells. Also, in addition to assessing the viability of the obtained endothelial cells, we analyzed the cell suspension for contamination. And according to our conclusion, the developed method makes it possible to get a pure cell suspension. After obtaining and assessing the quality of the cell suspension, we performed a series of experiments to simulate the transplantation cell suspension. The first part of the experiment was done on a trephant corneal scleral disc, and this slide shows the stage of staining Distemate's membrane with trypan blue dye to assess the primary condition of the endothelium. We know that when there is a higher density of endothelial cells, the blue stain will not be bright because the cells don't absorb the dye and only Distemate's membrane absorbs. Next, we remove the endothelial cells using a microtyper and an irrigation flow with an approximate diameter of 6 millimeters. After removing the endothelium, we restain the stemens membrane, and a bright blue color indicates the absence of endothelial cells on the surface of the stemens membrane. And this is how we modulated a potential patient with endothelial dystrophy. For transplantation of endothelial cells in suspension, we took 0.1 milliliters of this, and using the cells counter device, we found that this volume of suspension contains approximately 100,000 cells. For transplantation, we used a glass cannula because it is the safest for cells during their passing. Before transplantation, the suspension onto the corneal disc, we previously determined that 10% of cells are lost when passing through a glass cannula. After transplantation of the suspension, the corneal disc was left in an incubator for 4 hours, and after that time, the suspension was taken and the cells were recounted, and approximately 36,000 were obtained, which is 40% of the initial number. So the adhesion of endothelial cells in the experiment was approximately 54,000 cells, that is 60% of the initial number. Why did we take 100,000 cells in 0.1 milliliter suspension initially? We needed to fill an area of the stemens membrane with a diameter of approximately 6 millimeters with cells, and we know that the average density of endothelial cells in a healthy cornea is about 3,200 cells per square millimeters, and after the calculation we obtained the required number of cells. At the end of the first part of the experiment, we performed a scanning electron microscopy, and based on the results of which we determined obvious differences in the microstructure of the physically healthy endothelium and the one we transplanted. And this indicates that there are no primary endothelial cells of the corneal disc, and the cells that we see on electron microscopy are transplanted donor endothelial cells. The second stage of the experiment we performed on the cadaver eye. And first, we removed the epithelium from the cornea. After that, we marked the required diameter of 6 millimeters, and next, the surgical incision was made 2 millimeters wide. Using a Sinski hook, we removed the endothelial cells, trying not to damage the stemens membrane. And after this, we inject trypan blue dye to see the stemens membrane area after removal of endothelial cells. Next, using a glass cannula, we took 0.1 milliliter of a pre-preparated cell suspension and injected it into the anterior chamber of the cadaver eye. The surgical incision was sutured, and we turned the cadaver eye upside down. The cornea was placed upside down position in an incubator for 4 hours. And next we trypanated the corneal disc, and it was sent for cultivation for 7 days. We have to say that cultivation was necessary only to determine the availability and phenotype of the cells in the experiment. At the end of the second stage of the experiment, we received the results of scanning electron microscopy and immunohistochemistry, and transplanted endothelial cells adhered the completely closed the form defect, and the functionality of these cells is confirmed by the expression of two main proteins of endothelial cells. And in conclusion, we have to say that according to the circle formula and based on the minimum allowable amount of 500 cells per square millimeter, only about 15,000 cells are needed to fill a defect with approximately diameter of 6 millimeters. In the first and second stage of the experiment, 100,000 cells were transplanted into 0.1 milliliter of suspension, which, according to the results of scanning electron microscopy and immunohistochemistry, made it possible to achieve endothelial cells adhesion in the formed central zone of the defect. At the same time, in the research of Japanese scientists, 1 million cells were transplanted in suspension, but it was cultured cells. Considering the possible ways of cell loss that have not yet been studied, for example, at the implantation stage, or adhesion to the structures of the anterior chamber, migration to the posterior segment and trabecula, and etc. The further task of our study is to get the maximum number of endothelial cells in suspension to achieve the primary goal, corneal edema resorption. And thank you for your kind attention.